Hello, in this video we will discuss about the introduction to reporter gene assay. So what is the reporter gene assay? So reporter gene assay, it is used for identification of gene function in the patient and disease identification. So disease identification for the genetic disorder or the genetic disease like cancer, which that we can identify how their potential to transcribing and translation of the protein synthesis. So the reporter gene assay is used to signaling pathway, gene regulation structure of regulatory elements. So for example, here I will draw a cell as a sample cell when we will take sample from the patient with a specific tissue, for example, liver or other type of tissue. So it is basically the ligand will bind with the signaling pathway. So in this way, the basically the DNA here you can understand. So in this way here is you can understand the DNA and contain a sample cell in the nucleus, which that isolation of gene promoter, the promoter of interest, which that will be separate, but the function is that the promoter will trigger the reporter to transcription to translation of the protein, but the reporter is the similar luciferase enzyme producing reporter gene, while the promoter is basically promoter of interest, which that we will take as a sample to understand how they will work. So in this way, the transcription to translation to produce messenger RNA, and messenger RNA basically translation to produce luciferase enzyme. So the same mechanism, but 5' regulatory element is here. In this way, the potential promoters will be a 5' regulatory elements can be potential promoter, known promoters, and partial promoters and enhancer elements. So in this way, let's begin to understand the preparation of the plasmid, which that is recombinant plasmid after the taking the promoter of interest, which that promoter of interest take by the PCR amplification to from the patient gene as a promoter of interest, which that insert in the plasmid, as well as the reporter can be a luciferase producing gene. This reporter will also bind, and the yellowish color showing the 3' untranslated region microRNA recognition element MREs. Okay. So in this way, this is the plasmid. So this plasmid is basically insert is the transfecting cell. This transfecting cell will take the after the insertion into the transfecting cell. After the transfecting cell, basically contain in the plasma membrane or into the cytoplasm or into the nucleus. So in this way, here you can see this is the basically the nucleus contain a plasmid. This plasmid is basically contain a promoter and reporter and the MREs. So in this way, you can understand it produce the messenger RNA. It can be bind with the genome or into the cytoplasm. So remember, after this messenger RNA will go into the cytoplasm and translation to produce luciferase enzyme. So remember, so the strong promoter can be a more production of messenger RNA and the more luciferase enzyme production, while the weak promoter is basically less messenger RNA production and is equal to less luciferase. So the non-functional can be a non-messenger RNA is equal to no enzyme production. So in this way, the non-functional also for identification either promoter of interest is working or not. So here you can see the luciferase enzyme, which that will be convert the luciferin protein in the plant, but not in the animal cell. But the luciferin active to produce the color is the here green color. the bioluminescence light generating process. In this way, here you can see this is the MREs, which that is the microRNA. mRNA, which that inhibition of gene expression in animal cells. So that is why we are taking the specific cell for understanding better. So remember, so MREs is basically inhibit the messenger RNA for further production of luciferase. But in this case here, I am drawing again the promoter and regulator. Regulator is the basically the reporter and the different situation. For example, more production of luciferase, it means the promoter is strong. So identification, for example, gene, and luciferase when decrease, it means weak promoter, while if not produced, it means the promoter is non-functional. So we can identify the gene which that is we can get a promoter of interest to understand either promoter is functional or non-functional in the human cell for understanding the cancer cell proliferation or other oncogene activation. So oncogene promoter checking. So here is the luciferin sequence is basically specific promoter is required. So the specific promoter means the fibroblast cell will that promoter will produce, while the neuron will not, and epithelial cell will produce the color. So the advantage is of the extremely sensitive, producible, quick analysis. And the three luciferase for reported assays is Firefly, Ranila, Nanolog. So in this way, the application is gene expression analysis, promoter strength analysis, microRNA regulation analysis, and promoter structure analysis. So this is the application, and I hope you make sense about the introduction to promoter gene assay, which that is used for the cure of the disease and detection and identification of the disease like cancer or other syndrome or other a liver disease or other, for understanding what is going on in the cell and what is the activation of gene will produce that protein will damage the cell. So thanks for watching. Please make sure to subscribe, like and share. See in the next video. Bye.